Hi there, this is Dave Nguyen, CEO of Brain Scanology, and in this presentation we will talk about how Genie identified quantitative subgroups of lung alveoli. And I just want to say that though in this project we analyze the morphology of lung histology, the same analysis would work on tumor histopathology. For this pilot study, we analyzed images from this paper by Banerjee et al. They created an artificial lung in which they can sustain the lung of a mouse inside of a cage and gradually increase the amount of air pressure in the lungs, causing the alveoli to expand. In the bottom you see is a normal lung. As the air pressure increases, the alveoli expand and increase in shape. In the top row is a lung infected with pneumonia. And as you can see that as the pressure increases from left to right, only some of the alveoli enlarge and change shape. First, let's talk about how the LCPC transform within Genie works. In step one, you have the outline of the object of interest. And what Genie does is it finds the smallest bounding box or rectangle that touches all four sides of your object. The center of that rectangle will become the center of a radial grid of 71 lines. Genie then finds the intersection of every line and the edge of your shape as shown in step 3. And now it can calculate the distance of every intersection to the red dot, which is the origin. Now this object only has one edge, but if an object has multiple edges inside or outside of it, then all intersections on the same line will be added together into one number, such that Genie turns the data in step 3 into step 4, and then it applies the fast Fourier transform to the data in step 4 to derive what's called a frequency plot. And this frequency plot is a multidimensional representation of the shape that you began with. Multidimensional data is like the day, month, and year of your birthday. It's a multidimensional representation of your age, and you can calculate your age as a scalar value, that is the number of years that you've been alive. You can calculate that from your birth date. Your birth date contains much more information than just your age. Similar to this analogy of your birth date versus your age, you can then calculate a scalar value, like your age, one number to represent the shape you began with, by creating an index. A simple index could be to sum all the bins together into one number, or to derive a ratio, that is to divide the value of one bin by the value of another bin. In this way, that simple scalar value represents the shape that you began with. At Brain Scanology, we measure something called pure shape, which is very special to our software called Genie. So if you measure objects at the original scale, you cannot separate the effect of size from the effect of shape. But Genie allows us to resize these two objects such that they here have the same height. And when we resize them, we constrain what's called the aspect ratio, so the shape doesn't skew. And what we compare then is their shape apart from their size. We call this pure shape. And this is something only Genie can do. Let's quickly talk about how we extracted shapes. So here is an image, the alveoli in the lungs. Since this was a pilot project, we manually outlined the shape of each alveoli. In images that were complicated, like the infected lungs, we separated the image into 3 channels, one of which gave us clear edges of each of these alveoli. After we manually segmented each alveoli, we then isolated each into its own canvas. And then to measure what we call at scale, that is, we measure each object at its original size relative to each other. We rotated every object such that the longest internal line is now horizontal. And then we rotated that line to be vertical. And then we applied what's called the radial grid processing system as described earlier. So this is how we measure shapes at scale, that is at their original magnification relative to each other. One object is larger, one object is smaller. To measure something called pure shape, what we did was we again isolated each object onto its own canvas. We rotated such that the longest line is horizontal. But then what we did was we resized all objects to be the same horizontal width. So previously, the bottom object was smaller than the large one, but after we resize it, they both have the same width. And then next, we rotate these objects 90 degrees clockwise, and then we did radio grid processing. So no matter if we do measure things at scale or pure shape, each shape becomes a series of numbers. In this case, it's 71 numbers. And these 71 numbers are a multidimensional representation of the shape that you started with. In the same way that the month, date, and year is a multidimensional representation of a person's age, as opposed to how old they are. That's a scalar representation. These 71 numbers are a multidimensional representation of each shape. So since we have multidimensional data for each cell, we did k-means clustering in order to identify subgroups. And we also did principal component analysis on this data. And then what we did was we color-coded the principal component analysis plot according to the clusters identified by k-means clustering. And for k-means clustering, we use what's called the elbow method to approximate what is the optimal number of clusters identified. And for each of these groups that we'll discuss, 3 was the optimal number. Let's first look at the data from measuring these alveoli at scale. So here are the two-dimensional PCA plots, plotting PC1 and PC2. And we color-coded these groups according to their k-means clustering identity. So k-means clustering identified 3 groups in each of these conditions. And you notice that in the normal lungs measured at scale, there's a pattern which we call the diamond pattern, in which each cluster identified by k-means clustering, it takes on a specific location and shape. When we look at the infected lungs, they maintain this diamond pattern. And so this is evidence that there are quantitative morphological subgroups within the lungs of normal mice and infected mice. But then what we need to do is we need to validate that these subgroups identified by k-means clusterings are actually biologically meaningful. You see, because k-means clustering is a mathematical algorithm, and it almost always finds the existence of subgroups, which means that just because it can find subgroups doesn't mean those subgroups are actually real or biologically meaningful. So the way that we prove to ourselves that they're biologically meaningful is we find the middle 5 dots in each of these categories and we put them side by side, right? We find the middle 5 cells, we put them side by side to compare visually. Since we don't have any clinical data that goes along with these images, all we have is the shape of the cells. So when we do that, here we extracted the middle 5 cells of each cluster. And as you can see that here, this is the normal lungs, going left to right, increasing pressure, that there are 3 categories of shapes. You have the simple, small, circular shapes, and then you have these medium-sized, more complicated shapes, and ultimately you have the large amoeboid complicated shapes. And as you increase more and more pressure into the lungs, the alveoli expand into more and more complex shapes. Right here, we call this the butterfly morphology. So these are the fully expanded large alveoli in a healthy lung. Let's look at the cells in infected lungs. Right, so here, very interestingly, 3 groups still exist in the infected lungs. However, as you start increasing the pressure in the lungs, the smallest alveoli collapse. They become even smaller, right? Which is counterintuitive in terms of the mechanism through which pneumonia causes breathing problems, right? Not only do they prevent the expansion of the alveoli that can enlarge, they cause the small ones to collapse, which might be 2 distinct mechanisms through which pneumonia infection causes breathing obstruction. Very interesting. And as you can see here, that even though the smallest group collapses under increasing pressure, the other two groups continue to expand under increasing pressure, just like they do in the normal lungs. However, in infected lungs, the final complexity that they can reach under these 2 highest pressure conditions is less complex than in the normal lung, meaning pneumonia infection prevents the alveoli that can enlarge. It prevents them from enlarging into their most complex shapes that are observed in the healthy condition. Now let's look at pure shape. Previously, we measured the shapes of the alveoli at scale, which means we measure both shape and size at the same time. But Shape Genie allows us to measure pure shape, which is special because we measure shape apart from size. And when you look at the PCA plots and the distribution of the three clusters identified by k-means clustering, you see that the pattern is no longer the diamond pattern, but now we have a three-piece pie. And this pattern exists across all of the normal conditions, but also in the early pressure conditions of infected lungs. However, as you increase pressure in the infected lungs, very interestingly, the morphology reverts back to the diamond pattern here. So again, k-means clustering identified three morphologically distinct subgroups of alveoli shapes, even when measuring pure shape. Now let's look at what these shapes look like. So here we see that there are three distinct subgroups of pure shape. And first is these flattened elongated shapes. And then you have these more cuboidal squarish shapes. And ultimately you have the complex shapes, right? The butterfly shapes. And as you increase the pressure from left to right, you see that each of these categories of shapes begins to increase in complexity. All right, notice here that all these shapes have the same height. And the reason is because we're measuring pure shape, right? We resized every shape to be the same width before we rotated them 90 degrees clockwise. And we did that to separate the effect of shape on size. And interestingly, when we measure the pure shape of infected lungs, we see again that there is a collapse due to pressure, right, in these smallest category. Now these are circles, these objects are filled in. They do have a white space in the middle, but they're so small that our software algorithm accidentally filled them in. But as you can see, even when measuring shape apart from size, there are three distinct groups, and that pneumonia infection prevents the two larger categories from reaching their full level of complexity when compared to pure shape in the uninfected lung. So in conclusion, regarding what we found about alveolar morphology, first we found that there are three quantitatively distinct morphologies of alveoli in both normal lungs and infected lungs. Second, we found that the smallest category of alveoli actually collapse due to the combination of infection and increased air pressure, which might be a distinct mechanism of breathing obstruction compared to infection preventing alveoli from enlarging. Okay, and then lastly, pneumonia infection reduces the contour complexity of the enlarged alveoli, not just the size of the enlarged alveoli. Meaning morphology is likely a different mechanism of breathing problems as opposed to just reduced alveolar size. In conclusion, for the purposes of discovery, Shape Genie can quantify morphology that is hidden to the naked eye and traditional methods such as area, perimeter, average signal intensity. And it is great for discovering substages, subtypes, and transition states. And for those interested in AI and machine learning, Shape Genie can produce clean data that creates precise classification models that even deep learning cannot. Because deep learning relies on the user to define how many subgroups there are. So if the scientist doesn't know how many subgroups there are, deep learning doesn't either. That's the end of our presentation. We hope that Shape Genie will help you find amazing things in your research. As we like to say at Brain Scanology, it is now possible.